Hi, everyone, and welcome to today's Seed World Canada Retail Strategy webinar. I am Mark Zinkowitz, and I serve as editor for Seed World Canada. And today I'm happy to be your host. Now, today's theme is innovations in cereals. We're going to hear from three experts who will talk about their work in wheat and oats and how they're helping further both national and international development in cereals. Now, before we begin, I would like to thank our sponsor, CCAN, for their support. CCAN, of course, is Canada's largest supplier of certified seed. And so it's natural that they'd be sponsoring today's webinar talking about innovations in cereals because that is something that CCAN, of course, has a big stake in. And so thank you very much to CCAN. If you have a question at any point during our webinar, please just type it into the YouTube chat box and we will take your questions at the end of our webinar. You don't have to wait until the end. You can ask your question at any time, but we will tackle that as we near the end of the webinar today. This webinar is being recorded and will be available on our website, seedworld.com, in the coming hours. Now, like I say, we have three speakers today. Francois Utz is the National Science Lead for the Breeding Innovation and Crop Germplasm Portfolio at Agriculture and Agri-Food Canada. He's based in Lethbridge, Alberta. Welcome, Francois. How are you today?
Very good. Good afternoon, Mark.
Excellent. Good to see you, Francois. And Adele Jamalzai is a PhD student in plant breeding and genetics at Washington State University, working in wheat. Welcome, Adele. How are you?
Oh, thank you for having me. I'm great. Great to be here.
Excellent. And finally, Jim Dick is oat breeder for and owner of Oat Advantage, based in Saskatoon, Saskatchewan. And some of Jim's oat varieties are actually available through CCAN, which was another reason I wanted him to be on today. I know he's had an important connection to Canada's wheat breeding community for several years now. How are you, Jim?
Good. Thanks, Mark. Thanks for having me.
Not a problem. Thank you for joining us. Now, I'm going to chat with Francois first. So, like I say, I'm just going to remove you, Jim, and Adele from the stage here for a little while, but we will rejoin you in a few minutes. So, just hang tight. Francois, thank you again for your time. Now, our readers
My
might
pleasure.
be familiar with you because if they got our July edition of the magazine, either in print or they saw it online, you were on the cover of that magazine talking about AAFC's variety development in Canada, the work that you do in that regard, and talking about really about the future of that. And you were telling me all about how AFC determines its research direction. And for those who maybe went to the Seeds Canada annual conference in Edmonton back in July, you actually gave a presentation there. It was a virtual presentation, but you gave a presentation on gene editing in wheat. And this is something that is really cool that I've wanted to talk about for so long, gene editing just in general, but especially in wheat, because we've been talking for many years about things like, hybrid wheat and, oh, when are we ever going to see hybrid wheat? And we've talked about, GMO technology. And of course, there currently is no GM wheat available on the market, but that's kind of something else that people have speculated about. But here we are, from what I understand, that AAFC actually just recently planted its first gene edited wheat plots. This is something people may have read about on our platform, Seedwell Canada, or just in the media in general. And I'm curious, Francois, where are these wheat plots located? what does that look like, and what is the significance? I mean, this is a pretty big milestone for Canadian wheat farmers. So yeah, what can you tell us about where these are located, and illustrate for us what a milestone this really is?
Yes. Well, thanks very much, Mark, for the opportunity to talk about it. First of all, you speak about gene editing. This is a new technology that create a lot of expectation. We see a lot of potential in using these technology, and there are different products in that technology. We hear often about CRISPR-Cas9, but there are a family, essentially, of nuclease that could be used in plant or animal in order to edit genomes. And AFC is always interested in AFC scientists, always interested in exploring how these new technologies can be leveraged, help them either to answer a biological question or be incorporated in their breeding scheme in order to develop new trait. So indeed, we got our first first field trial with a wheat edited wheat line. And this project is led by Dr. John Laurie and André Laroche. They're both located in Lethbridge Research and Development Center. And so that field trial happened on our federal land, on our research land. And this was possible because we have some science experts, but also the support staff. So I want really to recognize the technical team has contributed as well. People who are mastering a number of technologies. I'm not sure all the audience understand the amount of skills that is required, the capacity also that is required in order to make this achievement. We certainly see more scientific publication reporting gene-edited trait in different commodities, but wheat is a little bit more challenging than some diploid plant species. It's hexaploid, so in that particular project, these two scientists and their technicians have looked at editing a couple of targets, gene targets, but they are present in essentially 6 copies. And so as they have in that project used CRISPR-Cas9. They had to master the delivery of that this technology in cells and tissue, a regenerate plant, and there is a fair amount of molecular evaluation of the genetic material generated indoors in control environment. And I had the opportunity to see these plants in growth cabinets, so very control growth environment, and I could see differences in phenotypes. And our science experts have recorded these various differences in the plant phenotype, such as flowering date, plant height, number of tillers, etc. But it's always interesting to see the plant in its true context in the field, because as we know well, you may modulate the function or knock out one gene in that case, and multiple copies in this hexaploid genome. You still have to observe this phenotype in the real environment. And so that was the purpose of running this first field trial. And our science experts have observed these plots, taken a lot of data, and again, seen some differences in phenotypic differences across a different derived line from this editing work. And I had the opportunity also to see it by myself. So it is a great achievement because it does require a lot of skills. And this work was conducted from the lab all the way to the field. It would require more, I would say, field season to analyze these lines, and keep in mind some of them would still be segregating because of the fact that these edits are not fixed yet, and there's multiple, I guess, copies or various number of copies of the targeted genes that have been edited. So yes, we're very proud of the success of our scientific team, and we're learning a lot also from this experience.
Yeah, Yeah, I bet. Well, and it is exciting because, gene editing is sort of the latest buzzword, especially in the headlines, right? It used to be, GMO was the big thing, and now you hear a lot about gene editing out there, and it's generally seen as a quicker and maybe a less costly alternative to GM technology. I'm just curious, Francois, what are some of the most, and I'll ask you to speculate a little bit here. I mean, nobody knows for sure, of course, at this point. Things are in the early stages, but in terms of some of the most promising traits that we could maybe one day see in wheat using this method, what could some of those traits be, particularly when it comes to things like sustainability, climate resilience? I mean, those two things are talked about so much right now. Do you have any thoughts on what we might ultimately see as a result of this?
Yeah, so when we think about application in order to further improve the sustainability of the Canadian wheat or other crops, as well as the climate resilience, what I'm thinking at is these abiotic stressors. What we're seeing is it's only in prairie region, but other part of Canada, there is period of drought. So there's limitation in availability of water, but there might also be some heat stress. You may suddenly have very high temperature, um, and for this cool season crop in particular, it is quite a challenge. The plant has the cultivar grown in Canada are not that well adapted to higher temperature for a longer period of time. And so gene editing could become a great tool to uh, target some very specific genes associated with how the plant responds to environmental cues. And that is, in general, looking at the transcription factors that modulate many different genes' response, turning them up and down uh, within the same 24 hours or over a longer period of time. And in fact, the work that John and Andre have done related to the adaptation to climate change and drought tolerance. So I think that's where there is a great opportunity. But we see also benefit just in expanding the genetic diversity of the domesticated crop. The great success of the breeder in generating varieties that are performing so well related to that extensive selection that they have done, and they so narrowed the genetic diversity within these plant species. So gene editing could be used to uh, uh, further or bring back a bit of diversity within this elite genetic material. And that's where the technology could be applied. Not, I mean, with there's still a knowledge gap and the need to acquire more knowledge about how these different genes are involved in the plant's response to environmental cue, but it could also result into um, uh, expanding that genetic diversity and providing essentially a new trait, or expanding the range of adaptation to temperature, to limitation of water availability.
Hmm. Yeah, it's just the possibilities are, pretty much, well, I don't want to say endless with this technology, but I mean, it's huge in terms of the potential of what could happen. And given that, it could take less time and money to develop a gene edited crop compared to the process, especially regulatory process that has to be gone through if you're creating a GM crop. Do you foresee gene editing maybe becoming like the preferred method for cereal development in Canada in general one day?
Well, I think it's a fantastic tool, and we see a greater investment going toward editing the genome of the host plant rather than looking at transferring genes from another species into the domesticated plant species. So certainly in the public research environment, I see investment going more to leveraging that genomic knowledge of the host crop species And certainly, you mentioned the regulatory challenge associated with GM. What is interesting in Canada and other jurisdictions, a great effort has been to clarify these regulatory guidelines and so create greater certainty, which is important to make a decision where to invest and what technology can be used. Understanding there's a number of tools, and gene editing is one tool in the basket of tools that breeders and other science experts working in that breeding continuum have access to. There's certainly also some, well, there's, I think, remaining challenge associated with the IP. Do we have the freedom to operate, to use this technology to develop a new commercial product? That was a major encumbrance working with these other GM technology. I think we have a greater latitude working in gene editing, understanding that some of these technology are quite relatively free of encumbrance. So it gives that possibility to use them and work from the lab again to the field and develop a commercial product. And they come with also significant advantage over the GM technology. Gene editing is a technology that is providing precision and accuracy. So we're editing what exists already in the genome with that precision, with very low off-target mutation created using this technology. And interestingly, artificial intelligence is even used to further improve the efficacy of these nucleases and reduce the off-target risk. So great potential to again mobilize the result of the massive investment that went into genomics, like sequencing the genomes, understanding how the genes are working together, the symphony that happened in the plant genome. It's still recognizing these gaps in our knowledge. And so the tool, the gene editing tool, would be great to develop new products, but also to accelerate the pace at which we fill that gap, knowledge gap.
Well, and like you say, products, that's important because when I interviewed you for the article that was on the cover in our July issue, I think I put the quote on the cover. Actually, your quote was that it's not AFC's job to just generate knowledge, but then to go the extra step and to ensure the what you call the impactful commercialization of that knowledge, and to ensure that that knowledge can then be used constructively, commercialized. And so are there any specific traits or innovations that AFC is most eager to see implemented in Canadian agriculture as a result of this?
Yes, definitely. And so we have to, I'll take you back to the strategic plan for science. It's a 10-year strategic plan for science where we want to mobilize our science capacity to further enhance the innovation solution to the great challenge we're focusing on. And you mentioned earlier the sustainability, the challenge with these extremes that we see, in particular in continental climate, so in prairie region. So we look at, and I should mention, keep in mind, gene editing is focusing on very specific target. The work of the breeder is to bring or to select genotypes that are going to meet all the merit criteria in order to be registered in Canada, but also to exceed essentially the merit of what is already available. And that's how farmers are seeing the next plant variety being better than what they have seeded previously. So we're looking at traits that could be transformative, that complement the ability of the breeder to select within the genetic diversity. And some of these traits related to or are associated, with very complex metabolic processes. So we're thinking at photosynthesis. We're thinking at nitrogen biological fixation. And these are two examples of very complex traits involve many genes, and in some instances, the genome is located in more than one place, and not just in the nucleus, but also in the organelle. And so it requires certainly a lot of skills, knowledge in order to work with the nuclear genome, but also the organelle genome. And that's where I think that the gene editing technology can complement the other basket of tools that the breeders and genomics experts have access to, keeping in mind that genomic prediction and selection or phenomic selection are also great tools available for accelerating the selection of new plant varieties. So this one gene editing could help focus on these complex metabolic processes where there's very limited genetic diversity within the plant species. And so again, photosynthesis, nitrogen biological fixation, I think would be the traits that we need to focus on.
Very exciting stuff. Thank you, Francois. We had a question actually come in for you, but I'm going to save that until the end. I don't want to tackle it right now. We will do our Q&A at the end. So hang on the line. I'm just going to have a little chat with Jim and then Adele, but we will join you again at the end. Thank you so much, Francois.
Sure. You're welcome.
Jim, hello. How are you today?
Good. Thank you. Thanks for having me over on your webinar.
Well, no, thank you for joining us. Now, Jim, like I say, you are owner of a company called Oat Advantage, and you've had quite a journey from starting, and you're based in Saskatoon, Saskatchewan, like I mentioned earlier, and you've gone from starting an oat breeding program with, I believe it was the United Grain Growers in the 1990s, to founding Oat Advantage in 2008. And I just want to show, you mentioned this is the story that you find yourselves in. This, I'm assuming this is your lovely family, and so
this is a while ago too, because I thought I'd take a picture when we all looked a little younger and cuter, maybe.
Well, I was going to say Oat Advantage, I mean, it really is a family affair. And so I was hoping you could sort of just tell us in a nutshell what motivated you, before we talk about some of the innovations that you've undertaken, what motivated you to shift to independent breeding after spending time with, larger organizations like UGG. I think you're also with Agricor United, and to kind of strike out on your own and say, hey, I want to be independent. I want to do this myself. Like that's not easy to kind of go it alone. Well, not go it alone, but to be an independent breeder. What made you want to do that?
I think that over the years you build up things, whether it's from education or early employment, and then research in these companies like UGG, Agricor United, and you develop a capacity not just for the things that but the people that you're working with. And over the years, the opportunity came to maybe when something comes to an end, to begin something new like this. And there's also was I would say a gap in the research that was taking place in oats. Oats is a small crop, maybe a little bit forgotten sometimes, but certainly there was an opportunity for a smaller player like ourselves to be to add value to the oat industry.
Yeah, well, and we're looking at some of your plots here, some of your oats, and so it's it might be a small family-owned breeding company, but I mean, there's nothing small about what you're doing. I mean, oats is, and we'll talk about this a little bit later. I mean, oats is actually becoming an increasingly popular crop in Canada. It's a hugely important crop. I eat a lot of oatmeal. I like to have my bowl of Cheerios before I go to bed. Like oats are a huge part of our food chain. And I'm just curious how you've managed to overcome some of the challenges that, maybe are typically associated with plant breeding. I mean, it's a resource intensive and a time consuming process. And being an independent breeder, how have you managed to manage that?
Yeah, the challenges are large, and the process is long. You definitely pull on, contacts and family and the resources you can to make it happen. You engage in other work to also pay the bills, contract work, and different things like this. And so, going back to the beginning, starting again in 2008, you just get going quickly, but in those early years, it is a small effort. I mean, you manage it together with other things, but it grows. But the challenges, I was familiar with how to do things, and so the challenges were not insurmountable.
Well, and like you say, you're familiar with how to do this, and that is why you have released new varieties to the market. I
Yeah.
mean, after years of work in breeding, I mean, we know it takes, you know, a decade, a dozen years from, the time the first cross is made to eventually
Yeah,
seeing
that's
that new
right.
variety come to market. And this has really paid off for you. In 2019, you celebrated the first commercial releases that Oat
Yeah.
Advantage had. You had ORE 3541M and ORE 3542M. What were the key factors behind the success of these varieties? And in other words, I guess, how did they stand out in terms of,
Yeah.
performance, in terms of, milling character, things like that, compared to other existing oats? What
Yeah.
what made these ones special?
Yeah, we were glad to have Secan pick these up and be a champion for us in the marketplace for providing these through their seed grower network, and that was really amazing. And we did find early on that the like in that picture just before there, the being in a seed grower field or whatever, and there are many of them, we would be we'd be able to work with and get knowledge from how things were going out in the field. And what would make them stand apart was something, I mean, that Secan saw some good agronomics and some good quality that was all there. We did come to learn too that the innovation that was part of these two varieties, are the 3541 and 3542, was surprising. And we had set out on a quality kind of a pathway, but these ones definitely set themselves apart for their ability to have very stable and high kernel quality, and other things, and good yield too.
So kernel quality, that's obviously something that the industry is looking for, and so I'm assuming like older varieties were maybe a little bit deficient in that area. Like how are these varieties in terms of kernel quality? Does it, for those who are maybe not familiar with it, does that make for better food, or what are the main
Yeah.
advantages of that?
I think, part of oats is, oats 100 years ago is a feed for animals and things like
Yeah.
this, and the bulk of it was very valuable for horses and things to pull machinery and things. Later on, I mean, we're a long way from that, and now oats have to go through many technical processes through large mills and things, and so the kernel quality or kernel uniformity that we had in those two, or we have in those two varieties, is really profitable and efficient for this technical machining, like machine processing. And so that was something that really, we need to leave behind just the oats for the horse and buggy. We're going to highly efficient in industrial, but really well done milling, and we see that varieties like 3542 and 3541M really beautifully complement the milling process.
Yeah, well, and as technology gets more
Yeah.
advanced, I mean, our lives are increasingly dominated by machines and now
Yeah.
artificial intelligence, and so as that happens, you need varieties that, yeah, like you say, are easier to process, that the
Yeah.
machines can handle better, because we're
Yeah.
living in this totally
And that,
different
yeah,
world now.
that next slide there, just a quote. I think that this is inspiring. We're looking dynamic breeding program, to target ambitious radical changes, and that's not to say, and radical changes can be actually small incremental gains at times, but we want to do more than just make little improvements. And so I think we were fortunate to have a bit of a leap forward, and that's a goal to not just improve a little bit, but to make some really significant changes in oats.
Yeah, well, and as Francois was talking about earlier with gene editing and wheat,
Yeah,
there's different technologies that are coming out now that are going to affect the course of breeding, and but yet we still have to go through. You still have to go through that process, no matter what technology you're using. I mean, you're still
yeah,
there's a lot of work from that first cross to the time it comes to
yeah,
market. And
absolutely.
can you just walk us through in a nutshell that typical timeline that you go through for developing an oat variety from the initial cross to the market release? Like, what does that look like?
Yeah, in those 12 years, there is that you said on, and that 12-year cycle, let's say, would begin potentially every year. You can start a new 12 years. So once you've gone through the first 12 years, you have many more coming along. And I threw up some pictures here. There's a picture of crossing, and that was my eldest daughter crossing there. And it's nice to take your children to work and things like this.
Well, and
But yeah,
sorry, I'm just going to interrupt you for a second. Fibonacci sequence.
yeah,
I don't know if anybody's read or seen that movie, The Da Vinci Code, but
right,
that was a part of that, the Fibonacci sequence with all the scrambled letters, and so
right, right.
that's kind of neat. Actually, I'm hoping you can, as you go through this slide, kind of explain to us what that is.
Yeah, there's a good book too called The Golden Ratio. But there's these plant patterns that, and I have a picture of a sunflower there because sunflowers are even just a very tidy way of looking at a pattern. And if there are kind of arcs in the sunflower that go one way, and there's another shorter arc going another way, but the number of arcs one way another form are part of a mathematical sequence. And in oats too, actually, just looking at that panicle there in the middle, like 1 1 2 3 5 8, those are first numbers in a Fibonacci sequence. And in the field, biologically, oats have to put up, like any plant, with a lot of pressures and things like this, and so patterns don't always follow what they might be. But our goal in the first years of breeding, like the first 1 2 3 4 years of breeding, you're looking at a lot of like patterns or new things that pop up naturally. Oats has a lot of variability within it. Some people call oats a messy hexaploid. I think with wheat, it's perhaps a tidier hexaploid. But so there seems to be lots of variation and even mutation that happens in the field on its own in the course of events. So we're taking advantage of that in the first 4 years, looking at plants and then drawing out ones that seem unique for one reason or another, and seeing what we can do from architecture to seed quality, looking for combinations that maybe formerly were maybe not thought of as being possible. Like if we think about plump grain with high beta glucan, those were somewhat thought to be maybe mutually exclusive. Maybe like even protein and high yield can be that way. But you're looking to break those, let's say, connections and try to get to the best of what we want. So the first 4 years of working on that, and that's in every project. And then as you move toward evaluation and going into commercial or cooperative testing, for instance, the Western Cooperative Oat Registration Trials, you're collaborating with many people in a public system that is thankfully coordinated by Agriculture and Agri-Food Canada, and they do a great job as a partner to undergird the system of testing and create like a really great opportunity for others to plug in. And so then in the years that follow up to years 8, 9, 10, you begin seed production, and then hopefully with a release to farmers by the year 11 or 12, let's say.
Yeah, it's definitely a long time, but it's as we all know, time does go by fast. And I'm curious,
Yeah.
just in thinking about the future, before I chat with Adele here and we
Yep.
do our Q&A at the end, what are some of the traits or the innovations that you're most excited to introduce into future oat varieties, especially in the context of Western Canadian
Yeah.
agriculture?
Well, if we pop through to the one graph slide that has a curve on it, or a number of curves, this one here, this is one of the things that popped up for us and that other larger industries, milling groups have come to see, maybe some of the things that we're seeing. What we're looking at is, the taller curve in the middle represents a clustering of some traits. So what we've been seeing is when a train load or a truckload of oats may look at the outset as if it was uniform, but really there's a lot of variety within it. And so we're trying to, instead of having a lot of range of sizes, we're trying to narrow the peak of and to create varieties that produce a very dense concentration of highly uniform oat kernels. And we did have that in our variety so far, and we're working to improve that. And so I did have this is kind of quirky, but it's like arts and crafts, but I'm just going to show up this one thing here. This is a little, what I call an oat rollout. So it represents kind of like a bar chart of grain off the combine. So the tallest one here, the most important one, is a sort of the value fraction of oats that we would like to call. It's about 67, close to 70% of a load of this. It's one of our varieties would be very uniform, whereas typically you see a much more spread out kind of situation. This is another bar chart of a typical variety, and the seed on the right here, I guess it's my left, but you're right. This is actually a fraction of oats that is not useful at all. And in this case, it has about 27% non-usable grain. So, and it always gets used in something, but it's not optimal. And so what we're trying to do is we're trying to create, repurpose the plant to create exactly what we want for the exactly right reason. And when we want to have that growing on the farm with high yield and high uniformity, then we would have something where all the fertilizer, the diesel fuel, the time and energy, the land, the rain, everything is brought together, not to produce suboptimal grain, like a wide variety of this and that. We're talking about manufacturing, and so we want a very tight outcome, and that is exciting for us. And in that picture too, that had that chart on it, there's also a little there's a little kind of electron microscope kind of, or maybe just a microscopic look at with oat kernel density. We're also looking at trying to repurpose a little bit of the way that the plant creates kernels to fill in the gaps, the creases, and the various shapes, so that within the same structure, the outer structure, we can fill the oat more to create a denser oat. At the same time as more of the uniform outer size, but the inner size of the package would be higher density. And I think about this. I've got a picture of 2 rail cars there. The rail industry created, the bottom rail car is a shorter rail car. It fits on the same tracks, but it carries more, and so we're trying to create an oat that can carry more within the same kind of outer structure.
Nice. I like that. An oat.
I think that's sustainable. That's because the more optimal you have it, the less waste of fertilizer and things, or waste of seed that you really didn't want, like off-target seed. So we're trying to have a lower carbon footprint for the grain that's produced.
Yeah, an oat that can carry a heavier load, so to speak. So.
Yeah.
Oh, that's really neat. Thank you, Jim. This is terrific.
Thanks, Mark.
And I'm just going to have a chat with Adele, and we'll do our Q&A. But thank you. Hang on the line, and we'll chat with you again in a few minutes.
Super.
Thanks a lot, Jim. Hi, Adele. How are you today? You might be muted still.
Sorry. Thanks for
There
having
we
me.
go.
I'm great.
Oh, well,
I was enjoying all the conversation and great research that Francois and Jim were talking about.
Well, and I want to talk about your research because it's really cool. You are a PhD student at Washington State University, and you are working in wheat. And I wanted to have you on to sort of bring some international flavor to our webinars. We're so far talking about Canada, but I want to talk a little bit from an international perspective. And now, can you I'm just going to load up your slideshow here 1 second.
Oh, great.
Here we go. Now you are enhancing spring wheat yield through integrating QTL pyramiding and high throughput phenotyping. So that just went a little bit over my head. So I was hoping you could tell us in a nutshell exactly what you're researching here and why it's important.
Sure. My research exactly centers on unlocking the full potential of spring wheat by, as you know, by combining the QTL permitting with high throughput phenotyping. So what we did is that we strategically selected and stacked some QTL. When I'm saying QTL, I mean genes that control important yield traits like the size of the grain of wheat, like this number of spikes and kernel weight. So we are targeting different QTLs, as you can show in the next slide. These are the QTLs or gene that we selected to combine all of them together. As you can see, like WAPO is for increasing spikelet number that was found in UC Davis, California, and QTN-MSD6B for boosting tiller production that was in Montana State University. So we selected these QTL genes, and by stacking these genes in one variety, we are aiming to create wheat plants that are genetically, optimized for maximum yield. But at the other side, the high throughput phenotyping, the cool part is that the real game changer is that how we're using high throughput phenotyping. We're flying drones, technically, equipped with advanced sensor to collect real-time data. And real-time data on the plants that we already stacked the genes in together. And we want to examine their performance in the field, and we want to track some traits like spike number, plant height, and plant vigor, and also plant health. And with the help of deep learning and also with high throughput phenotyping, we wanted to record other yields related traits like the number of spike, as I said, or the plant lodging. And yeah, that's it. And I hope that this technology actually give us instant feedback in how these genetic improvements are translating into actual yield.
Wow. Yeah, well, and it's fascinating because it's, yield is such a complex trait that's influenced by a lot of factors. But, wheat yields over the decades have gone up so much. I mean, there's been so much great work done in terms of wheat yields. And what are some of the biggest challenges that you face when you're trying to increase wheat yields?
Oh, that's a great question. If you ask any plant breeder what are the challenges, they just can talk about that for hours and hours. So yield is what we call is a quantitative trait. Quantitative trait means it's controlled by many genes, and each genes have a small effect. That's where the complexity comes in. It's not just a single gene that you can tweak in to increase yield. It's a combination of factors working together, and they don't always cooperate nicely. So another challenge is that's the main reason for my project is the compensatory effect. What is it actually? In wheat, improving one trait like grain size might reduce another trait like grain number. So a lot of studies before, they found QTL or gene that increased the grain size, but when they could transfer it to another, variety, they didn't see any, changes in the yield. So that's the reason they don't see any, changes in yield. So this happens because the plant has a limited amount of resources. And when you increase one yield component like grain size, it often draws away from another. It's like you're trying to make a bigger pie with the same amount of ingredients, and you can only increase the size of one slice so much bigger, other slices get smaller. And also, on top of that, environment also play a huge role in these traits. So a QTL or a gene that works very good for yield in one location, say under optimal water and temperature condition, might not perform the same in a harsher or more variable environment. So we are always working to balance the genetic potential of wheat with real world condition. So the complexity comes from the fact that yield is just not one thing. It's like a system. It's a whole suite of trait interacting with each other and also with the environment. So the whole thing and the whole goal of my project is we have to balance all these factors to increase yield without causing, the trade-off that I was talking about.
Yeah, well, and like you say, looking at some of these technologies here, like the RGB imaging and some of the
Yeah,
things that you're able to analyze using this technology, I mean, it's really amazing. And I mean, you've got a slide here on deep learning,
yeah.
and so some of the technologies that you're using to discover some of these things like are really incredible. The amount of data that you must generate and have to make sense of must just be mind-boggling at times. And so I'm just curious, like, what impact you see this research having, say, on global food security in the long
Um,
term. I mean, you've got all this data now that you've got to bring down and figure out, like, where do you see this ultimately building to?
Uh, I mean, this research, as you see, it's a combination of genetics strategies and also high throughput phenotyping and, AI and deep learning. So, technically, most of the traits in the field are doing by hand, and people are just walking in the field and recording some traits, but we are just trying to do all of it automatically with drones. And we have different types of drones in our lab. And the thing that I'm doing that's kind of like an innovation in my project is that I'm trying to use drone as like an RGB, like red, green, and blue, as you know, to record some traits that are not actually recorded by drones before, like counting the spike of wheat in the field by deep learning. Like I'm trying to, train a model and then annotated each image and see, okay, these are the traits. And then the model trained on the, detecting the spike, and then we can have like in the number of spike in the field without like walking under the sun and stuff. So I see this research as playing a huge role in the future of food security. As the population grows, especially in regions where agriculture is kind of thin, the pressure is on us as plant breeder to produce more food with fewer resources. By developing high yielding wheat varieties, we are essentially giving farmers a tool they need to produce more wheat, even in challenging conditions. This could help reduce reliance on import in like third world countries or like, many region, and making countries more food secure. And the broader agriculture industry, the techniques we're using, like QTIL permitting, as I told you, and high throughput phenotyping with the help of deep learning, are advancing how we approach plant breeding. It's all about precision and it's all about food security. And in long term, I think we'll see more crops that are smartly bred for both yield and sustainability, which will be crucial as we face more, unpredictable growing season, due to climate change, as we all know how it's going to affect us in the long term.
Yeah. Well, in referencing your slide here, wait, what was the question again? I imagine you feel like that sometimes when you're talking about some of these technologies, and it's just like thinking about, where this is all headed. What Francois talking about, gene editing in wheat, and then applying some of these technologies that you're talking about. It must be pretty mind-boggling to you sometimes to imagine like where this could all go at the end of the day, what it all means for yield and different traits. It's the sky, I guess, is the limit, as they say.
Uh, I mean, the genetic research into agriculture has brought significant advancements for us.
Yeah.
As a great talk Francois gave us about the genetic gene editing, I really believe and hope that genetic research is redefine agriculture sustainability. as climate change intensifies and resources like water and fertile land becomes scarcer, genetic research will be key in developing crop varieties that thrive with fewer inputs, less water, fewer fertilizer, and reduced pesticide for all of us. And also, at the same time, they maintain high yields. So, genetic research in agriculture is on the brink of an even greater transformation over the next decade, I guess. In my opinion, the future looks incredibly exciting. One of the most promising development in the rise of precision-based breeding technology, I believe, is going to be CRISPR-Cas9 and gene editing. And we can introduce beneficial traits like higher yield or better stress resistance in a fraction of time that it would take using, traditional breeding method. It's just not about speeding up the process, though. It's all about the precision that will allow us to target complex traits like yield and all the challenges and problems that I told you about yield. Yield is a pretty complex trait.
Yeah, exactly. No, thank you so much, Adele. This is fascinating stuff. I just want to bring Jim and Francois back onto the stage here so we can do a quick Q&A. And again, if you have a question, just type it into the chat box. And Francois, I just want to start off with you. We had a question come in regarding CRISPR-Cas9 gene editing. And so this comes from Eliza asking, I was referencing GMO earlier and how gene editing and GMO are two different technologies. And Eliza was asking, how this CRISPR-Cas9 process is different from GM. And so obviously, from what I understand, Francois, with GM technology, you are introducing what's called foreign DNA from a different species into a plant. With gene editing, that's not the case, correct? You're simply working within the plant's own genome, flipping switches within that genome without introducing anything that wasn't there before, correct?
That's correct. The gene editing tool essentially, it starts by having these nuclease that can cut the double-strand DNA, and we cut it at a very specific site. So that CRISPR-Cas9 would be designed to recognize a very specific sequence, induce that cut, and it would cut as long as the sequence is available. The nucleus, the DNA machinery will repair, but it would be cut again as long as the nuclease is available until the repair introduce an error. And that's what we called insertion or deletion of a few base pair. Then you have a mutation created at a very specific target site. You didn't introduce any foreign DNA. And the goal is not to have any residual DNA that could be associated with the CRISPR-Cas9 or other nuclease or sequence coding for these nucleases. So yes, it's very different in that sense. There is no DNA sequence from another species that would be introduced in the targeted plant using gene editing.
Yeah, and some people will still say, well, you're still tinkering with the genome, but it's important to note that plant breeders have been altering genomes for thousands of years. I mean, it's anytime you make a cross, or which people have been doing since time immemorial, very, very long time, you are altering the genetics. And so I guess you can think of gene editing as a way to simply a more direct route, I guess, of doing that more conventional plant breeding, I guess.
Yes, so it's very specific, very targeted, and it gave that precision. So if we talk about the genome of wheat, it's huge. The CRISPR-Cas9 nuclease would be designed to target just one of the essentially a very small sequence. So it is indeed very targeted, and introduce targeted mutation. At the end, what you have is a mutation. And mutation do occur constantly in the genomes of living organisms. It occurs also from generation to generation. If we were to look at mutation within the offspring, we would find them, and they are not present in the parents. So it's from one generation to another introduced low rate of mutation. And there's other technique that have been used for more than 100 years to induce mutation. So certainly radiation, other chemicals that induce mutation. But in that case, they create random mutation across the genome, and then it's a bit of a mess. But breeders have used this to, again, create more genetic diversity. And whether it's in grapes and you look at a different wine type, well, they vary for one mutation. And it was created naturally because mutation do occur in every living organism, including us.
Yeah, well, that was fascinating for me when I first started working with Seed World about, I guess it'd be 11 years ago now, and I first started learning about plant breeding and things, and so-called conventional plant breeding for many decades now, like you say, has used radiation to induce mutation. You soak seed in various chemicals to induce mutation, and so this is all so-called conventional plant breeding, and so it's there are many different ways of doing this, and I guess it just depends on how we think of it in terms of, well, this is foreign DNA to create this, but with gene editing we're not introducing foreign DNA, and there's many different ways to induce it. And I guess, Adele, for you, like if you think of some of these technologies that you're using, like drones, for example, and I mean, you're using technologies that people could never dream of before. I mean, to have drones and all these different ways to assess, the different qualities of plants. And so I guess by extension, that's just another way of helping to induce these, these genetic changes in these plants. And so I guess right from day one, human beings have been altering genomes, whether we're doing that directly in a laboratory, through gene editing, or you're doing it by crossing two plants together, or you're using multispectral imaging or a drone to look at plants, or in some cases inside of a plant or inside of a seed. And I'm curious, Adele, what you think in terms of, we were talking earlier about global food security and the role you see genetic research playing in global food systems. I mean, will we eventually, Adele, get to a point where, the kind of research that you're doing right now, it's seen as really groundbreaking, and it's like, wow, but I suppose, Adele, we're going to get to a point where your research and what you're looking at just becomes sort of just commonplace, I guess, just the way of just the way that it's done, correct?
Mm-hmm. Yeah, exactly. Like high throughput phenotyping, as you said, is a pretty advanced and new technology that we use right now, and it's a pretty cool tool in plant breeding too. Like as I said, gathering data required a lot of manual effort, and but with drones, we can have some data that actually is not accessible to our naked eyes. So, like drones like Inspire, DJI Inspire for multispectral imaging, as you said, it is equipped with sensor that capture data across multiple spectral bands beyond just what we see in the visible light spectrum. And also a little bit on a multispectral imaging for those who probably are not familiar with it, is it actually allows us to assess plant health and vigor in a ways that, as I said, we can't see it ourselves. For example, a healthy plant reflects more near infrared light compared to a stressed plant. By analyzing different bands of light, we can calculate various, vegetation indices that are directly related to yield. Like it is before even we are harvesting it, with the data, with the large data, as you said, we can predict like, okay, this variety is not good for yield, so we're not going to work on that anymore based on the drone imagery and the data that we got from there. And on top of that, we can also use like RGB imagery, and RGB imagery are also good for, as I said, recording some traits that are, when we record traits in the field by persons or people are doing it, we all know it's going to be a little biased. So if I go and record some trait in the field, if you go, probably they're kind of be a little different together because we're like humans and it's subjective. But with drones, we know that all the data that we got is kind of like very, all the same. And I'm really excited about both drones that we are working here, because we can monitor the traits real time, not just at harvesting. And it just gives us deeper insight in how the plant are actually responding to environmental stresses. So I guess the combination of what am I doing, like the high throughput phenotyping and QTL permitting, like genetics and also advanced technology, gives us a better perspective on like how to increase yield. And as we know, yield is a pretty important and complex trait for plant breeders, and it really helps us to tackle some challenges for food security, because we know that population is growing day by day.
Yeah, and it really gives you more control, as Francois mentioned. You don't have to induce all these mutations and all these plants and then go out and pick, and you can figure out, okay, this is exactly what I want to do. So I can go in and I can do that. And then, Jim, I'm going to give you the last word here, Jim. This question is a little bit over my head, but hopefully you can tell us the answer. What specific, and this is for you specifically, Jim, what specific crown rust resistance genes are you using in your varieties? For example, PC94 or PC91, are you also working with quantitative resistance, aka adult plant resistance?
Well, there you go. No, I won't be the expert on that, but we do again depend on Agriculture Agri-Food Canada, because they put out some very nice varieties, and then we can share sometimes and do crossing back and forth. And so some of the, I forget which genes are in the one, which PC94 or whatever that we have, but we cross with varieties that have the leaves that we like under stress, and so we do evaluations over the years and pick and cross with what we have. And so then we just we don't, as a sort of a smaller operation, we don't necessarily do that kind of screening, or sometimes we partner with others who can, but so for crown rust resistance, we rely on just breeding good varieties together.
Excellent. Well, thank you so much, everybody. This has been terrific. And I just want to thank our audience today for spending your lunch hour with us. This webinar was at 1 o'clock Manitoba time, but probably be lunchtime in Alberta. And so, yeah, thank you to those of you who took part of your lunch to join us. I want to thank our sponsors, CCAN, as well, for their support. A recording of this webinar will be available in the coming hours, so check our website, seedworld.com. Adele, Jim, Francois, thank you so much for your time today. This was fascinating. This has been, I think this has been one of the best, most exciting webinars I've done in a while. Just the technologies and the gene editing and wheat, the stuff, Adele, that you're doing with the QTL, the high throughput phenotyping, is amazing. And Jim talking about oats. I know, according to statistics, Canada, actually, there were more oats planted this year, Jim, than there was barley in Canada, according to the latest statistics. And so this is definitely a good news story for cereal breeding in general. So thank you to all of you. Thank you to our audience. This is Mark Zinkowitz of Seed World signing off. Have a terrific day, everybody.
Thank you.
Thank you, Mark. Goodbye.